Welcome back, my dear students. Today we are once again together to study another interesting topic. This topic is control of blood glucose concentration. This topic is from unit Homeostasis of GCE A level. This topic will help the students of Cambridge, edexcel, AQA and OCR, and they will learn how blood glucose concentration is maintained and how diabetes and different symptoms of diabetes reflect. So in this discussion today, we will discuss about how blood glucose concentration is maintained in the optimum range, and if this is not maintained and it remains high, how this leads to the development of diabetes mellitus, its types, and how we can counter this diabetes and we can detect the blood glucose concentration. So let's start with our discussion today. As you people know that glucose is the primary requirement. It is taken as the primary respiratory substrate by all the cells in the body that has to be transported across the body by bloodstream. So blood must have a particular concentration of glucose in it all the time, so that continuous supply of glucose shall be maintained throughout. So where the blood glucose is obtained, where the glucose is obtained in the blood, so that is normally obtained either through digestive system, but its prolonged supply is maintained through the liver particularly. So that glucose in the blood travels throughout the body, and wherever it is needed in the body cells, it is taken up, and its uptake may be increasing or may be decreasing depending upon the energy requirement of the cells or the tissues. Normally it is assumed that in the blood at an average of 100 milligram per deciliter cube glucose should be available, or its range is 80 milligram to 120 milligram per deciliter cube. This is the optimum range. But this has been observed that while after a meal this can increase above 120, or during starvation time or meal skipping time it may decrease below 80 even. But remember, things go normal, life activities go normal if the blood glucose concentration remains within its optimum range, required range. So what happens if the blood glucose concentration goes high? Remember one thing, when blood glucose concentration goes higher above 120 and it remains higher above 120, so that is going to cause some serious problems. What are those problems? The primary thing is that when blood glucose concentration increases, it also increases the concentration of blood, and blood osmotic potential will decrease, resulting blood will fetch water from the tissue fluid, from the blood cells, from the cells that are lining the wall of arterioles. Now, when the cells are losing their water to the blood plasma, so resulting they are getting crenated and they will lose their working potential. So if they are the white blood cells, they will be weaker in their immunity power. If they are the red blood cells, so oxygen transport may be affected. If the cells lining the wall of arterioles, they lose water to the blood plasma, so they will cause shrinking of the blood vessels, and lumen diameter may decrease, and it can also result in reduction in the elasticity or flexibility of the arterial walls. Similarly, when the water is taken up from the tissue fluid, so tissue fluid in return takes up water from the tissue cells. So tissue cells are getting crenated, so they will also lose their working potential. On the other hand, when there is an excess water in the blood plasma, so that will increase the hydrostatic pressure, that will result higher blood pressure, and higher blood pressure can lead to stroke or other severe problems. Similarly, if blood glucose concentration goes below the normal range, again the very first thing that can cause a problem, that glucose supply to the various body tissues may reduce. Ultimately, energy production, so energy supply, respiratory activities, that can go reduced in the various body tissues. Okay, and on the other hand, reduced blood glucose concentration that may also lead an increased osmotic potential of the blood, causing serious consequences as well. But remember, there is a homeostatic control in our body, and that is homeostatic control that is maintained by pancreas. Pancreas in our body that is serving as gland, and it is serving as endocrine gland as well as exocrine gland. As exocrine gland function we are not going to discuss here, but as endocrine gland we are going to discuss because it is concerned with the homeostatic control of blood glucose. As endocrine gland, it has a tissue that is known as islets of Langerhans. This tissue contains two cells, alpha cells and beta cells. Alpha cells are responsible for the synthesis and release of a hormone known as glucagon, whereas beta cells are responsible for synthesis and release of another hormone known as insulin. Both of these hormones are produced simultaneously by these cells, and they are released into the blood in order to maintain glucose concentration within the optimum range. So these cells, with the help of their receptors, they can detect the glucose concentration in the surrounding tissue fluid and accordingly manage with the release of their respective hormones. Now we are going to discuss the situations where the blood glucose concentration may rise or may decrease. Now, one thing we are going to discuss is hyperglycemia. That means blood glucose concentration starts to rise or it becomes increased. Why it can happen? So, intake of sugar-rich or starch-rich meal may result increase in the blood glucose concentration. So, in that case, if the blood glucose concentration is going above 120, so both of these cells respond to the glucose concentration in the surrounding. Alpha cells will respond by decreasing the concentration of glucagon, whereas beta cells in the pancreas will respond by increasing the secretions of insulin hormone. Insulin travels through blood and it reaches liver cells, muscle cells, and cells of the adipose tissue. Insulin binds with its specific receptors on the liver cell surface membrane and on the muscle cell surface membrane. And due to the influence of insulin, increased uptake of glucose is started in these cells, and metabolism of glucose at an increased level that also initiates. Now, when the insulin binds with the receptors on the liver cell surface membrane, on the muscle cell surface membrane, that influences the opening or availability of special transport proteins known as GLUT transport, GLUT, transport proteins that are responsible for the glucose transport within the cells. Now, in the liver cells, there are present GLUT2 proteins, GLUT, GLUT2 proteins. These are the transport proteins that allow glucose to enter the liver cells. Whereas in the muscle cells, these proteins are not available in the membrane, but rather these proteins are available as membrane-bound vesicles or GLUT4 protein vesicles in the cytoplasm. When the insulin binds with the receptor, so these GLUT proteins containing vesicles move to the cell membrane, merge with the cell membrane, and add these GLUT4 protein channels in the membranes of the muscle cells. And that will start the intake of glucose down the diffusion gradient. When the insulin binds with the receptor, so there is activation of series of enzymes. For example, phosphorylase enzymes, kinase enzymes, they get activated. For example, kinase enzyme gets activated first. That is responsible for converting glucose to glucose phosphate, and this phosphorylated glucose cannot escape out of the cells. Why? Because the cell membranes do not contain any transport protein that allows glucose phosphate to move in or move out of the cell. So this is something to trap the glucose that has entered the cells. And this is not only a trap that glucose is trapped inside the cells, rather it also activates, or you can say also energizes the glucose, so it can easily be metabolized. Certain other enzymes, they are also activated, and these enzymes may either lead this glucose toward respiratory metabolism in order to release energy in the form of ATP and in the form of heat, or this glucose, phosphorylated glucose, can be metabolized to the synthesis of glycogen, and that will be stored within the cells. I repeat that GLUT2 proteins are present in the membranes of the liver cells, but their working is initiated or stimulated by the interaction of insulin with its receptors. Whereas GLUT4 proteins are not present in the membranes of the muscle cells, rather they are present as membrane-bound vesicles within the cytoplasm. But when the insulin binds with the receptors, so these vesicles merge with the membrane and add these GLUT4 protein channels with the membrane. Whereas in the brain cells, GLUT1 proteins are present, and these transport proteins are always open channels, and they always keep allowing glucose to enter the brain cells. So means brain cells are always respiring on glucose. Now this insulin also interacts with the cells of adipose tissue, and it stimulates them to stop the release of fat molecules in the bloodstream, so that the tissues are the organs that are respiring on fats. When the fat supply is cut down to those tissues or organs, so they also start respiring on glucose. So for their increased energy demand, they will take up more glucose from the blood, and this will result glucose being reduced in the blood. And glucose will reduce in the blood, and ultimately it will return to its normal level, in the normal optimum range. But remember one thing, that this is not means when the blood glucose concentration is decreasing back to normal, but it's not the end that the cells will stop taking glucose. No, cells will keep continuously take up glucose from the blood because of their respiratory needs. So blood glucose may start decreasing. If the blood glucose concentration starts decreasing, that time, now again both of these alpha cells and beta cells respond to the decreased blood glucose concentration. Now this time alpha cells respond by increasing the concentration of glucagon, and beta cells will respond by decreasing the concentration of insulin. Increased glucagon will travel through blood and will interact with the adipose tissues and will interact with the cells of the liver, and it will bind with the glucagon receptors. When this glucagon interacts with the glucagon receptors on the liver cell surface membranes, so that initiates a cell signaling effect. G protein is activated, that in return activates an enzyme that produces secondary messenger, that is cyclic AMP, and that spreads in the cytoplasm of the liver cells, and consequently, ultimately, it initiates, or you can say, a series of enzymes are activated that ultimately result in the release of glucose from the ends of the chains of glycogen molecule. And this released glucose is immediately released out of the cells down the diffusion gradient and through tissue fluid. Ultimately it reaches the blood plasma, so that blood glucose concentration may be restored to normal. At this time, when blood glucose concentration is reducing, it is also ensured that stress on the glucose should be reduced. So glucagon, when it interacts with the cells of adipose tissue, it stimulates them to release fats in the bloodstream, so that the body tissues or the muscles that can respire on fats, they must take up fats and respire on fats, and their stress over the glucose uptake should be reduced. And this is done in order to ensure that continuous supply of glucose shall be maintained for the brain cells, because brain cells can only respire on glucose, but various other body tissues and organs can switch over to fat or protein metabolism in harsh circumstances or in emergency circumstances. So this is how both of these hormones being produced in a coordinated concentration all the time by the cells of pancreas, and they keep helping and they keep maintaining blood glucose concentration in the optimum range. And this is what we have learned so far, that how blood glucose concentration is maintained homeostatically by the negative feedback mechanism with the help of pancreas, with the help of two cells in the pancreas, alpha cells and beta cells, that produce insulin and glucagon. But there are certain circumstances where we can find increased blood glucose concentration all the time in some persons. And this particular condition is known as diabetes, or what we call diabetes mellitus. That is a disease, that is a disorder. So diabetes mellitus is characterized by increased blood glucose concentration without any known reason, and that increased concentration is maintained for prolonged time, for all time in the blood. So this could be caused by various reasons. First of all, before going to discuss various reasons, we must discuss the two types of diabetes mellitus. One type is known as type 1 diabetes or insulin dependent diabetes. And the second type is type 2 diabetes or insulin independent diabetes. In case of type 1 diabetes, also known as insulin dependent diabetes, as the names suggest, that it is due to lack of insulin in the blood. So means this particular type 1 diabetes patient is not producing any insulin in the blood, or may be producing very little insulin. Now this could be due to certain reasons, like this could be genetic, means in some people it could be inherited from the parents, and the genes responsible for insulin production, they may switch off at some particular age, particularly at the age of 40. Similarly, as the aging process goes on in certain people, so in that case, telomeric depletion may also result in the development of type 1 diabetes. Now, telomeric depletion, what does it mean? In certain beta cells that are dividing again and again mitotically, and the telomeres are not maintained, so gradually one gene is being depleted at every consecutive replication, and that is not being transferred to the upcoming daughter cells. So at some particular age, when the newly developing daughter cells, they do not receive the genes to produce insulin, so they will not be producing insulin in future. So that will result initially in the reduced concentration of insulin in the blood, and in the further later ages, it will result a complete stop of insulin production. Similarly, if the gene responsible for producing receptors, glucose receptors that are present on the membranes that detect the high glucose concentration or low glucose concentration, if the genes for the production of these receptors are not transferred to the upcoming daughter cells, so these cells will not produce any glucose receptors, so they will not be able to detect any glucose concentration in the tissue fluid, so production of insulin will be affected. In certain other cases, autoimmune response, the faulty response of our immune cells, may also result no insulin production. In this case, what happens? That the faulty immune cells, they produce antibodies against glucose receptors, considering them as non-self antigens. So when these faulty immune cells consider glucose receptors as non-self antigens, they produce antibodies against them. Antibodies bind with the receptors and block them and inactivate them, or you can say deactivate them, and they can no longer detect the glucose concentration in the surrounding. So in that case too, no insulin will be produced because the cells are unable to detect any glucose concentration in the surrounding. In case of type 2 diabetes, that is insulin independent diabetes, insulin production is not the problem, but insulin cannot cast its impact. Means it cannot interact with the liver cells, with the muscle cells, and it cannot make them to increase uptake of glucose and metabolize it. And why this is so? This could be due to non-availability of the insulin receptors on the liver cell surface membrane or on the muscle cell surface membrane, or this could be due to autoimmune response, that antibodies produced against these receptors by the faulty immune cells block these receptors and do not allow insulin to bind with them. So that's why insulin cannot cast its impact. So blood glucose concentration can go higher than normal all time. So in both of the cases, how the patients are going to respond. In case of type 1 diabetes, the patients need external intake of insulin. They can collect from the stores, they can get consulted by the health authorities, and a particular dosage of insulin is suggested to them, and even they learn injecting insulin by themselves. And even as now we are having the more advancement in this field. So insulin inhalers are also available in the market with the modified insulin that can diffuse through alveoli in the blood, or even certain insulin can be injected that can stay for longer in the blood, in the body, and it can manage glucose concentration even for months. So this is also possible. Along with the insulin intake through external sources, these type 1 diabetes patients must regulate their diet. They should avoid sugar-rich food, and they should take small portions of meal with short intervals. Instead of taking bulk meals with long intervals, they should take small portions of meal with short intervals. That will maintain their energy level throughout, and they should go for exercise, walk, and it will reduce their blood glucose concentration, and it will keep their blood glucose concentration in the normal range. Whereas the patients of type 2 diabetes, as they don't have any insulin production problem, so they do not take and they do not need any insulin externally, rather regulating their dietary chart with the small portions of meal, with the small time intervals, avoiding sugar-rich meal, and at the same time doing exercise can help them maintain their normal blood glucose concentration. Now the question arises how one may know that he might be suffering from the diabetes. So there are certain symptoms that reflect in the patients who suffer from diabetes mellitus, and the symptoms are almost the same for both, whether one is suffering from type 1 diabetes or type 2 diabetes. Symptoms are the same. Like the patient of diabetes mellitus feels a frequent urge of urination, means he produces large volumes of urine and he needs to urinate more frequently than the normal person. At the same time, in the urine of that person, there is high glucose concentration detected, as well as high concentration of ketones and the mineral ions is also detected. These patients also feel hungry within shortest time, means they need to take meals, they feel hungry very quickly, and at the same time they feel thirsty very quickly and they need to drink a lot of water with short intervals. At the same time, there is another symptom observed, that these patients start experiencing weight loss and body thinning is observed in these people. Now let's discuss that how these symptoms are appearing, reasons for these symptoms. You know that insulin actually stimulates liver cells and muscle cells to take up extended glucose. Without insulin, these cells do take glucose, but they do take glucose for their normal needs. But when insulin is there, these cells take up a lot of glucose and they store that glucose in the form of glycogen. And when there is a time between meals, so means during that time when there is no external intake of glucose, so that glycogen is gradually hydrolyzed to release glucose, and that meets up the body demand. But in case of diabetes, there is no glycogen reserve formed within the cells. So the glucose that enters the body through meal can survive for 2-3 hours, and after that glucose supply gets reduced in the blood. And as there is no reserve, so glucose supply is very much reduced, and blood glucose concentration may drop very steeply. So in that case, the brain immediately creates a sensation of hunger, so that a person must eat something immediately and must eat something a bit sweet or sugar rich in order to immediately manage with the glucose concentration in the blood. So that's why a person feels hungry. When there is high glucose concentration in the blood, as it is not converted to glycogen, so most of it is filtered out in the glomerular filtrate, and it is less reabsorbed. More is retained in the filtrate and very little is reabsorbed into the bloodstream. So renal filtrate or the glomerular filtrate that contains large concentrations of glucose in it. And when there is high concentration of glucose in the urine, so water reabsorption and mineral reabsorption is also reduced, and particularly at the loop of Henle. So more of the water and more of the mineral ions are retained within the renal filtrate, and they are passed out of the body along with urine. So this is the reason when the urine contains large volumes of urine as well as high concentration of glucose and mineral ions as well. So that's why, as there is a shortage of water and shortage of mineral ions in the body, so that creates the body being dehydrated, and a person feels thirsty and needs to drink a lot of water in order to compensate for that. When there is no reserve of glucose in the body in the form of glycogen, so in harsh times or in times of need, body muscles, body tissues, body cells, they switch over to fat and protein metabolism. So in that case, when they are respiring more on fats and proteins, so that will result reduction in the fat contents in the body, a reduction in the protein contents in the body. Remember one thing, we do not have any stored protein, but the protein that is used is the cellular proteins that is normally for the functionings of the cells. But that protein is used, and that fat stored as adipose tissue or around the organs, so that fat is also used up. So ultimately, reduction in the fat contents and protein contents make a person grow thinner, and at the same time weight loss is also observed. And as fats and proteins are metabolized, so ketones are produced, so that's why the ketones are reflecting more, keto acids are reflecting more in the urine. And when there are more ketones being formed, so these ketones in the blood, they can reduce blood pH. So this reduced blood pH, lack of glucose in the blood, ultimately will result a person even experience coma. So this is going to be very fatal in that case. So that needs to be maintained. So in order to avoid such sort of circumstances, a person need to keep a check on his blood glucose concentration. So if there is a person who is suffering from diabetes mellitus and he has experienced all these conditions as symptoms, must immediately consult to a doctor and must get the appropriate treatment. At the same time, he has to keep a check on his blood glucose concentration. How he can do it? Nowadays we have got a very advanced form of dipsticks, that is the glucometers, or you can say biosensors. The biosensors are actually electronic devices. These electronic devices are actually containing inside transducers, and a strip, electronic strip, is used in order to drive these devices. That electronic strip has got a immobilized pad at one end. that pad contains enzymes, and a blood drop is introduced on that pad. Immobilized enzyme on that pad react with the glucose in the blood, convert it into gluconolactone and hydrogen peroxide, and other enzyme on the pad breaks down hydrogen peroxide. This creates a small amount of electric current, and a small circuit on the other end of this strip transfers this small amount of current into the device, where transducers convert that current into digits and amplify it and display that glucose concentration in the form of digits on the screen. So this is the very accurate blood glucose concentration reflecting just 5 seconds ago. Means 5 seconds ago you took a drop from your blood and introduced on that strip, and within 5 seconds that gave you the reading of blood glucose concentration. So this was about glucose, maintaining glucose concentration in the blood with the homeostatic control mechanism. And in this discussion you also learned about the diabetes mellitus, symptoms arising due to diabetes mellitus, and how one can keep a check on his blood glucose concentration. Stay tuned for further updates. Thank you very much.